So my name's Lynsey, and I'm a postdoctoral researcher in statistical genetics. So I spend my time much like a suspicious housewife, rummaging through people's genes in search of answers. So the work I'm going to talk to you about tonight is actually stuff I did for my PhD, which was analysing the genetic basis of depression in the Scottish population, which is where I'm from, quite a good population for studying such traits. And the way we do this is we get two groups of people, and so we have the first group, which is like the super sad people, otherwise known as cases, and then we have the other group, who are a bit more kind of the ever sort of encroaching prospect of the existential abyss doesn't bother me, pal, I'm fucking telling you, eh? Otherwise known as Scottish controls. But when we did look at these people and we sort of compared their DNA sequence in a sort of elaborate game of spot the difference, we didn't actually find anything. So part of this is because you're actually looking at about 9 million differences, and so it's more like a sort of depressed Where's Wally, except there's been a printing error at the factory and there's no Wally or Wanda or Odla or Woof or Wizard Whitebeard. So why is this? Maybe it's just because I'm crap at science. Lots of other people have tried and also not found anything for a long time, and the main reason is because we don't actually know how to measure depression. Which is a bit unfortunate, really. I wish someone had told me that before I signed up for my PhD project. But even if we did properly know how to measure depression in humans, there's still a whole heap of other problems. So, you know, if you're wanting to make a new drug to treat an illness, then you need to be able to do this in animal models before ethically you're allowed to do it in humans. And so this is often the rat, and you know, the way we measure depression at the moment, it's not a blood test or a stool test, although I wish it was. It's more of a sort of qualitative questionnaire. And this doesn't really work with a rat. You can't have a rat on its back in a chaise longue in a cage. You're asking it things like, do you often have feelings of worthlessness and guilt? Do you still take a pleasure in your day-to-day activities? Do you ever think that the other rats know what you're thinking and are plotting against you? It doesn't really work. So what can we do? So we need to find these tests that can kind of work in animals and humans. And so one example of this is something called cognitive bias, which is basically where you get like a good stimulus and a bad stimulus and an in-between stimulus. And what you find out is that people who have mental health problems tend to take the in-betweeny stimulus in the wrong way. So if I were to come up to you and say, that's an interesting top you've got on, regular you might be like, do I know you? I'd be like, no. They never do. And you're like, all right, well, thanks, I guess. Cheerio. Whereas depressed you might be a bit more like what you're saying, like you're starting a fight. Like, yeah, I'm just saying it's an interesting top you've got on. But again, you can't really do this with animals, can you? You can't really mince into an animal housing unit with rows upon rows of rats in cages and be like, sup y'all, nice jackets! Got one at the back eating its babies, but that's got nothing to do with what you've just said. So this brings me on to some work I did before my PhD with the illustrious rat boss, as I used to call her, kind of like Splinter from Teenage Mutant Ninja Turtles. And she devised this like pretty cool setup that was had a sort of start chamber and a sort of tunnely bit, an end chamber. And the end chamber had a couple of bowls of sand in it, and the little sort of should not be doing that gesture. My mom's going to see this on the YouTube. Anyway, the tunnel is lined in sandpaper, and we used different grades of sandpaper. So the rats would learn that if they were walking on rough sandpaper, they'd get to the end and there would be chocolate in a bowl. Sweet. They'd go through the tunnel with the smooth sandpaper and there'd be Cheerio in a bowl. Not so sweet, but better than a kick in the teeth. And so we train them, we just nail them through the maze over and over again, pop them in at start. Rats, are you ready? Gladiators, are you ready? Get through that maze, find some treats. And we make sure they weren't using their nose by doing the occasional blank trial. Now, I've never done animal work before, but I'll tell you something. If you've got a hungry rat and you put it through a maze and there's not a treat at the end, they are not fucking happy with that. There's a lot of cleaning up to do after that. Stand, not rats, not rats. But anyway, what we did was we had some of the rats were stressed, some of the rats were not stressed, and we got an in-betweeny sandpaper. The idea being, if they were feeling a bit more optimistic, they'd pick out the rough and get to the end and be like, oh, I think there was a chocolate. That's what I'm taking from that in-between grade sandpaper. And otherwise, I go through and pick out the smooth and be like, oh, I guess this is Cheerio, but fuck it. And this worked like quite nicely. But I don't know if any of you have worked with animals before in a clinical setting. It's not particularly what's the opposite of thankless? Thankful job. And you know, it can take a long time to train them. You're at their sort of behest. You might not get your lunch anytime soon. So I decided to write a song about this to the tune of Kanye West's Gold Digger. That's called Bull Digger. Let's have a look.
Yeah.
Jess is anticipating this wildly. She loves the trials at rapid speed. Yeah, she's the kind of rat I need. Yeah, she's a bull digger, and her results look good to me. Oh yeah. I ain't saying she's a bull digger, but she ain't hitting the response trigger. Her results into this figure. Use a pass to make the sample bigger. Learning trials day after day, trying to find the right way. But when she digs in an empty bowl, she's all looking at me like, what you playing at home? Told you want to chocolate like a Cheerio. You don't give it me now, I'm going to learn real slow. Pretend I don't know my way around the maze, even it's 586 trial of the day. Pick me up, I'm going to squeak, I'm going to batter your gloves and make you feel like a freak. If you treat me right and try to hide your frustration, I might learn the trials within the 16-week duration. Trials at rapid speed. Yeah, she's the kind of rat I need. Yeah, she's a bull digger, and her results look good to me. Oh yeah. We're up in wireless now. But like I say, that was before the PhD, so over the PhD and the postdoc and everything, I've been working more with human DNA, my favourite. And this is, you know, it's all good, but what you don't realise is just the sheer quantity of data that you get and how much sort of computational and statistical modelling you need to make sense of it all. And this is generally fine. There's a lot of people in my field who are computational scientists or statisticians. There's also a fair chunk of our demographic that, like me, are biologists. What? Got one there. And this is a bit problematic because I don't know if anyone has done a biology degree, but you tend to get assessed on things like the ability to accurately depict a snail's mouth under a microscope. It's called a radula, by the way. Or also measuring the length of your thigh, the length of a locust's thigh, and challenging the locust to a long jump in the name of physics. Which is probably about as close as a Scottish person has got to winning the Commonwealth Games. But anyway, this doesn't really prepare you for the fact that when you land at your desk and you're told that you're going to be using the Linux operating system, or as I like to call it, the nihilist choice, or using multivariate models to sample like population and mixed family data, doesn't always work that well. But I'm 5 years deep now, can kind of do it, and if I may, I would like to finish on a song that I've written to the tune of Less Than Jake's The Science of Selling Yourself Short. Now, I normally change these names to have a bit of a pun in them, but I think it's quite apt, to be honest. So, I've been spending my time on the hunt for mental genes. Not those ones from Topshop, though I'll confess they're pretty ream. Depression, they're the ones that make you sad, make you lose your interest in any hobbies that you had. But the problem with depression is that it's not one disease diagnosed by questionnaires based on subjectivity. I'll take your blood, analyze your DNA. Let's try again, didn't account for sex and age. And one more time, really think this family tree don't look quite right. You're your mother's second wife. I could be an expert, except I don't know any stats. What the F's a beta, I'm a nova, and if bad, use my 50s textbook, but the maths looks quite insane. Maybe try some websites before equations melt my brain. But the problem with using Wiki to look up terms that you have heard, it becomes Google Inception, looking up every second word. I'll take your blood, analyze your DNA. Let's try again, didn't account for sex and age. And one more time, really think this test statistic don't look right. I hope that I write up on time. I really hope that I write up on time. You're still your mother's second wife. Thank you very much.